Hello all, good evening. This is your biology teacher Alka Rajamon from GoScore. We are the fastest growing edtech in GCC and our aim is to provide quality education for all. Yes, welcome back. Welcome back. I hope you all still remember this phase. I hope you all still remember what we are doing today. Yes, this is another energetic session of your Heredity Chapter 8 of Biology. And again, this is me from GoSco. We are going for the YouTube live class. Yes, this is part 2. I hope all my students are here. I hope everyone is here. Hi all. Hello all. Yes. Hi. Hey, hey, hey. Yes, welcome. Welcome back all. And yeah. So I hope you all doing good. All are preparing for your pre-boards, your terminals. I hope you all are in a rush. But still, here we are to help you with the free live classes which can contribute to your board exam marks, your pre-board exam marks. So use these sessions. And as I'm always telling you, whatever you are studying, whatever you are learning, don't just put it in your short-term memory. Always convert it into your long-term memory by keep revising it, keep challenging your brain. Whenever you learn something new, don't just put it in rest. Always take a quick test for it. Always challenge your brain so that your brain will keep it safe forever. Yes, so revision, practicing, challenging your brain is the best option which is needed to keep your short-term memory in a long processing. Yeah. And I hope you all remember the last session. We were discussing everything about your chapter Heredity, right? We have discussed the terminologies in genetics. Mendel's contributions are yet to be done. Monohybrid cross, dihybrid cross, laws in sex determination, it's all still pending. So yes, today we are going for it. And yeah, do you remember the last comment which was in our class? I know that you all were so active in the comment session and we had a lot of doubts and queries in the in the comment session. And one of those comments were like, if I take a heredity test, would I be able to know my future? Right? One of you asked like that. And yes, I have to say the answer this life. That's how I entered my previous life, right? So I have to start my life with the answering. Heredity test is a test which helps you to understand the genetic composition of your body. Okay, if you're an adult, it is easy to take your blood or your saliva and understand the constitution of your blood or your saliva, the cell inside it. If you take the cell out of it, you will get to know what all genetic constitution are there. If there is any sort of issues in your gene, if there is some sort of bad mutations which has happened, can you be ill in the future? Is there any cancer causing genes which can get activated in the future? So yes, if you take a heredity test, you can definitely know your future. That is what it is for. And if you are a pregnant woman, you normally know about this amniocentesis, right? What is amniocentesis? Amniocentesis is nothing but you take the amniotic fluid from a pregnant woman and you check the amniotic cells in it, which will reveal the genetic constitution of your baby. So you can even predict a baby which is not born yet. So not only in an adult, even in a fetus which is still developing in a mother, you can predict the future, its health, everything can be predicted. Yes, that's the answer for the question. And thank you all so much for joining. I hope you all will be there supporting me until yeah, for 55 more minutes, I guess. So let's use the time to its best. Let's begin the session. I'll just quickly go through whatever we have learned. We have learned what is a trait. We have learned heredity, variation. We know the variation is maximum in sexual reproduction and minimum in asexual reproduction. We know the differences between genes, alleles, phenotype, genotype, inherited and acquired trait. And today we will be discussing a completely new topic, which is Mendel's contribution. So who is this person? Mendel. You have any idea about this guy? Please comment. You have any idea about this guy? Gregor Johann Mendel. Any idea? So this is known Okay. No idea, no idea. Nobody has any idea, I guess. Okay, so Gregor Johann Mendel is he was a great scientist who was a geneticist, who was a botanist. He has done a lot of experiments with the pea plant, with a lot of other green stuff, and he has actually brought in a lot of conclusions which we are using today. Even in 2024, we are discussing this name in heredity, in genetics, because this guy has contributed a lot to the world of biology. So we'll see what all contributions he has done. Gregor Johann Mendel was born in 1822 and he died in 1884. He is known as the father of genetics, and the plant he worked on was Pisum sativa, which is also known as our garden pea. And he has proposed three laws in genetics. It's also known as laws of inheritance, and you can also call it as principles of inheritance. So there are basically three laws proposed by Gregor Johann Mendel. It's known as laws of inheritance or principles of inheritance, which we'll be seeing in the upcoming sessions. Yes, hi all. Thank you so much for joining. Yes, hi. Okay, now Gregor Johann Mendel was a great botanist, great scientist. And why did he choose this garden pea plant to do all the experiments and to make such great conclusions about it and to make all these universal laws of inheritance? Why did he use a garden pea? Why such a plant? So there are plenty of reasons for choosing such a plant. A few among them are, and you have to keep these points in your mind. You might get a question from this area. Availability of detectable contrasting characters. So what is this detectable contrasting character? What does it mean? So garden pea is a plant which has alternative character. If there is a white flower, it also produces a purple flower. If there is a yellow colored seed, it can also produce a green colored seed. So all these traits, all these characters are so contrasting, so alternating that we can see the difference with our naked eyes. You don't have to go for a heredity test, or you don't have to cut open the plant and see what all differences are there. So if you need to study the character of that organism, you just have to look into the plant. Just looking into the plant, just that looking is enough to understand what all genes are there inside the plant. So those contrasting traits or contrasting characters are the first reason for choosing such a small plant with a very low lifespan. Yeah, that's the second point, which is short lifespan. Short lifespan means the garden pea would grow and die within months, right? If the seedling comes out, it can grow, yield and die within few months. So in 3 months or in 4 months, you can actually study the entire life history of that plant. So normally, whenever a geneticist or a biologist who's studying genes is choosing a plant or an organism, we always choose something which gives birth to young ones very quickly and die very quickly, so that we can study the entire life cycle in a very small period. I hope you understood it. Always choosing an animal or a plant which is having a very short lifespan is important in genetics, so that you can cover up the entire life history of the organism in a very short period. You don't have to wait. What if you take a human to study the genetics? You will have to wait at least 70 to 80 years, so that you can cover up the entire life history. So this is having a very short lifespan, which makes it easier for the geneticist to study that. And the third one is self-fertilization and cross-fertilization. So yes, these garden pea plants are bisexual flowers. It is easy to make cross-fertilization in those plants, or self-fertilization. They have both male and female organs inside them, which makes it easy to do both the fertilization, which helps to do the monohybrid cross and dihybrid cross. We'll see. So that's another characteristic which makes the pea plant more user friendly, I have to say. And then the fourth point is large number of seeds. So as you know, pea plant is not producing a single nut or a single seed, right? It basically has a long pod, right? It basically has a long pod. Inside the pod, it keeps these seeds, right? It has the seeds inside this long pod. So in a single fruit itself, there are plenty of seeds to study the next generation. So that's another reason, large number of seeds. Now, this is a question which was repeatedly asked, that is, what are the 7 pairs of contrasting traits in pea plants studied by Mendel? What are the 7 pairs of contrasting traits? So, this question might come as a saying, or it can twist, it can be twisted, or it can asked in several ways. So, you basically have to study all the 7 characters that Grigor Johan studied, and you have to keep those characters and its contrasting characters in your mind so that you can answer any question from that part very quickly and easily. Okay, so let's see what are the 7 pairs of contrasting character. So Mendel used Pisum sativum, which is also known as the garden pea. Am I right or not? Yes. Oh, hi. Hello, let me quickly go through the comments. Hi, hello, hi. Hi everyone. Hey Blessy, Alia, yes, hi Alia, and Sonia, Pooja, Alma, yes, Thomas John, yes, hi Basil, hi, hello, hello, hello. Thank you all so much. Thank you so much. Thank you so much for being here with me, being so supportive. Yes, be there, be there. Okay. So Mendel uses a number of he used a number of contrasting traits. Now we'll study which all they are. So the plant was Pisum sativum. Keep the name, keep the scientific name in your mind. If you are lucky enough, you might get 1 mark from that. So Pisum sativum, he chose these characters to be studied. Let's study the characters one by one. Okay, so the first character he took and studied was a stem height. Second is flower color. Third is flower position. Fourth is pod shape. Fifth is pod color. Sixth is seed shape. Seventh is seed color. Now, these characteristics have their alternative forms, which also you have to keep in mind. Okay, so let's see one by one. First one is stem height. The garden pea plant can be tall or it can be dwarf. So I hope from this you understood what I just said. They are contrasting traits. Either the plant is very tall or it is very dwarf. There is no intermediate stage there. So you don't have to get confused between the phenotypic expression of the plant. Either the plant is tall or it is short. Simple like that. They are contrasting traits which are easy to identify with your naked eyes. Understood? Okay. Hi. Yes, hello. Okay, so the flower color, that's the second trait, right? Flower color is the flower can either be purple, the flower can either be purple or white. It can be purple or white, completely contrast, right? It's a contrasting color itself. You don't have to get confused. And then flower position. Flower can be axially placed. It can be axial position or it can be terminal position. What does that mean? What does that mean? Axial flower. Suppose this is a plant. This is how the leaves are, right? So in axial position, what happens is the flower would come out of the stem. The flower would come out of the stem. This is how the flower position would be. That is axial. From the axis, the flower occurs, which is a dominant trait. Okay. And there is terminal position. Terminal, the word itself says terminal. What is terminal? In the case of terminal, the flower would come only from the tip of the plant or the stem of the plant. I hope you understood the difference. Axial means from the axis, terminal means at the tip. Okay, these are the two types of flower positions. Then we have the pod shape. What is pod? I just showed you what a pod is. What is that long fruit which is coming out of the pea plant? This pod shape can be different. It can be inflated. Inflated means what? A pod is inflated means what? Any idea? A pod is inflated means nothing else. It is just flat. It is just flat. No other shapes are there. Just flat. So, oh, I'm very sorry. Okay. So pod shape can be inflated like this. It can be inflated like this, or it can be constricted. Constricted. What is constricted? If there are ridges around the pond, ridges, you might have seen these beans and peas and all like that. You have seen the flat ones or one with the constrictions, the ridges. Okay, so these are the two types of pod shapes available. Then we have the pod color. Pod color is green and yellow. Green or yellow. The pod can be either green or yellow. Here in pod color, green is the dominant color and yellow is a recessive color. Okay. So in the case of pod color, green is dominant and yellow is recessive. Okay. Then going to the seed. Seed is the thing that is inside the pod, right? This seed can be yellow in color or green in color. But in the case of seed color, yellow is dominant and green is recessive. Understood or what? Understood or what? In the case of pod, the pods are always, just imagine our green peas or imagine the beans, the French beans and all. Normally, the beans are green in color, which is a dominant trait. Okay. But in the case of seed, it goes the other way. It's the other way. That is, seed color, yellow is always dominating and green is always the recessive one. Just the opposite. And seed shape, seed can either be round. That's the seed we all have might seen, right? That's a very common shape of the seed. It can be round or it can be wrinkled. It can be wrinkled. There can be wrinkled seeds in the case of pea. So it can be round seeds or it can be wrinkled. Okay, so these are the 7 traits of pea plant which were studied by Gregor Johann Mendel, and you have to study it too because you might get some question. Maybe if you're lucky enough, you might get some question from here. And always keep the contrasting traits in your mind too. Okay, done. Clear with everything? Any issues as so far? So far? Who is this gummy? Hi. Hello. I think I can guess that person, but I'm not guessing. Okay, this is a question which was asked before, that is, give the pair of contrasting traits of the following characters in pea plant and mention which is dominant and which is recessive. It's a simple question. You can easily say it. Comment the answers, please. Please comment the answers. I think you can easily say the answer. First character is yellow seed. What is the contrasting trait of yellow seed? Any idea? That is definitely a round seed, right? Okay, fine. Now you have to answer which is the dominant and which is the recessive one. Give the pair of contrasting traits of the following character in a pea plant and mention which is dominant and which is recessive. So in a pea plant, in the case of seed, yellow is, complete my sentence. Yes, yellow is the dominant trait. Yellow is the dominant trait and round seed is also a dominant trait, right? So if this is the dominant trait, you have to answer the recessive traits too. For yellow, which is the recessive trait? Yellow seed is a dominant trait. The contrasting trait, that is, green seed is its recessive form. Green seed is the recessive form. Going to shape of the seed, round seed is a dominant trait and wrinkled, yes, the wrinkle seed is a recessive trait. I hope you understood the concept. Okay, so they just have asked this question from what we just learned. All those 7 characters and their contrasting, alternating traits. That's it. Okay, moving on. This is another question from the same topic. Very simple. The characters that are not studied by Mendel in pea plants. So if you should know which are not studied, you should be knowing which was studied. Right? We know that 7 characters were studied, which we have already by hearted. So no issue. Just give me the answer. Can you please answer this question in the comments? Seed shape and seed color. Was it studied by Mendel? Yes. Pod shape and pod color. It was also studied. Yes. Flower position and flower color. Was it studied? Yes. Pod shape and pod length. When did he study pod length? No. So that's the option. Pot shape and pot color was studied, pot length was never studied. So that's the answer for the question. So these are the simple questions you might get. You might get from that small topic. So just keep those 7 characters in your mind now. I hope there is no issue until now. So I'm moving on with the next topic. We will get into the major portion of this chapter, which is none other than inheritance of genes. Yes, in the chapter heredity, you have to learn inheritance of one gene, inheritance of two genes. You will be studying monohybrid cross and you will also be studying dihybrid cross. So shall we move on? Yes, yes, Asma. Hi. Yes, hello. Okay, okay, okay. So, inheritance of one gene. So, this is where you're basically studying how a gene is transferred from one parent to the offspring, what all laws of inheritance take place in there, and all those stuff, which is very simple. If you are able to understand the concept that I'm saying, the questions you have to answer will be very simple. Okay, so let's see. Inheritance of one gene, it is also called a monohybrid cross. A monohybrid cross. Yes. Inheritance of one gene is studied with a monohybrid cross. And what is a monohybrid cross? As the name itself suggests, it is mono, where only one character is studied. So in a monohybrid cross, you basically take one character of any organism and learn how that character is moving from a parent to an offspring, or from one generation to the next generation. Simple like that. So one character. And in monohybrid cross, we basically have to do 2 steps. Simple like that. You just have 2 steps, and if you know these 2 steps, the cross is done. Let's see which all the steps are. So the first step is cross fertilization. Cross fertilization. So I hope you remember what I said before. Cross fertilization. Okay. So I hope you remember what I said before. Why Gregor Johann Mendel choose this garden pea plant? One of the reasons were he was able to do the cross fertilization and self fertilization in the same plant, right? That's the reason. So here comes, when we are doing a monohybrid cross, the first step is doing a cross fertilization. Cross fertilization is nothing but the fertilization is between two different plants, right? You're doing it between two different plants. Yeah. And then we have the self fertilization. The second step is, okay, the second step is self fertilization. Fertilization. Okay. Okay, now let's see what these steps are and how to do the monohybrid cross and what are the results of the monohybrid cross. Now I need your attention. I need you to focus in the class. Do not zone out. Do not go anywhere. Stay there. We are going to learn. We are going to learn the most important point. Okay. Are my students talking in the comment session? Why? I think you're supposed to talk to me, not between yourself. Come on, focus in the class. Come back, come back. They're having a huge chat in there. Okay. So monohybrid cross, it's nothing but a cross where only one character is studied. Okay. So in order to study the character, I'm going to take a very simple character, which is height of the plant or height of the stem. Okay. That's the character I'm going to study. And in order to study the height, which is the chosen character here, height of the plant or height of the stem can actually be, okay, height of the character or height of the stem can actually be in two different contrasting forms, right? If I'm taking height as a character to study, it can occur in two different forms. It can be a tall plant or it can be a dwarf plant, right? That's how it is. So height can be tall or dwarf, right? Yes, Kasi, so nice to see you again. Good. Yeah, hi. Yes. So height, it can be tall or dwarf. So let's see how to make this cross. Okay. So when you're doing this cross, what we are basically doing? First, we are doing a cross fertilization. Simple. Second, we are doing a self fertilization. So to do a cross fertilization, now I am taking two different plants. One is a tall plant who should be homozygous dominant, which should be crossed against a dwarf plant which is a heterozygous recessive. Understood? There is a tall plant and its genotype should be capital T, capital T. And the dwarf should be small t, small t. That is how it is, right? There is no other option for dwarf. Okay. So now let's make the cross and study how to do the monohybrid cross. Okay, let's start. When you're making a monohybrid cross, first you have to take the parental generation. First, the parental generation, which you can either denote with the capital P or you can literally write parental in words, no problem. Okay, so I'm taking the parent, which is tall parent against the dwarf parent, right? The tall parent would be capital T against small t, small t. In the second step, I have to take the gametes which would come from this parent from the homozygous tall parent. Since it is homozygous, yes, okay, okay. Since it is tall parent, the gamete would be single type of gamete. It's a homozygous dominant. So the only possible gamete from that parent is capital T. And when you're writing a gamete, put it inside a circle. When you are writing a gamete, put it inside the circle. I repeat, that circle, you can take your score away if you don't draw it. Yes. And then here, the only gamete possible is small t. Simple like that. I hope you got it. Got it? Yes. 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 Be active in the comments and calm down. Yes. Okay. Now we have taken the gametes. You just have to do the cross fertilization. You are making the cross and making a zygote which should later become an offspring. So the F1 generation would basically be capital T, small t, which is a heterozygous dominant offspring. That means how many offsprings you get in the first cross, in this cross, they all will be tall plants. They all will be tall plants, but not at all like their parent. Not at all like their parent. The parent was homozygous dominant, but the babies, the offsprings in the first generation is going to be heterozygous dominant. Anyways, the phenotype would be tall. Capital T or capital T small t. Phenotype would be tall. Simple. So F1 is denoted here to say that it is filial generation 1. What is filial generation? Filial means the offsprings. That's it. It is just to denote that it's the filial generation 1 because we are going to make one more generation, then it will be F2. Okay, so that's the first step, cross fertilization done. Over. Now we are moving on to the next step. That is, yes. Hi, Ephraim. Hi. Yes. Coffee venda. No, I don't need. Thank you so much. Okay. Yes. So now we are going to do the self-fertilization. Okay. We are going to do the self-fertilization. Okay. So let's see how to do the self-fertilization. I hope you all are drinking your water and coffee and tea and chilling in there. Just stay hydrated, but focus to my class also. Okay, focus. Okay. Now the second step is self-fertilization. So we basically have to take this F1 generation and cross it with another F1 generation. Simple like that. So let's see how to do that. See, we are taking the parents. The parent would be capital T, small t into capital T, small t. Gamete would be, since it is heterozygous, there are the possibilities of two gametes. Two gametes. We have capital T and small t gamete against capital T, small t gamete. Okay. Now I need the F2 generation. To make this F2 generation, I can simply use a square which is called a Punnett square, right? We have heard about this Punnett square, have you or not? Have you or not? Have you heard about the Punnett square? No, never? Okay. So Punnett square is nothing but it's basically a square where you can easily do your genetic crosses. The genetic crosses would be so easy if you are using a Punnett square. So we'll do it inside a square and let's see how to make that square. So now we have done with the cross fertilization. We have got the F1 generation, which is capital T small t. In the second stage, we are crossing the capital T small t with the capital T small t, which will be giving the F1 generation. Now let's see what the F2 generation look like. Okay. In order to understand what the F2 generation look like, I have to make a Punnett square, which should basically be a square inside which I would be denoting the gametes. I would be denoting the gametes. So let's see. In the first row, you basically have to divide it into such a way that one will be female and the other would be male. Okay, one will be female and other would be male. In the case of plants, we have no idea who is male and female, but we simply denote it in such a way that one set, the first row is for a male and the first column is for a female. So let's see how it is. Now, which all gametes have we received here? We have got capital T small t from one parent and capital T small t from the other parent. So, let's see how it is. In the first row, you will be writing capital T small t. In the first column also, you will be writing capital T small t. Simple like that. So, if you are doing the Punnett square, it would be very easier to get the results. You don't have to get confused. Not only for monohybrid cross, even when you're doing a dihybrid cross, when you're using the Punnett square, it will be very easy to come to the conclusion. So it's basically capital T, which is a tall baby, capital T small t, which should also be a tall baby, capital T small t capital T, which is also another tall baby, small t, which will be another dwarf, not another, the first dwarf baby. Got it? This is how we do a Punnett square. I can even do that there also. Hi all. Hey. Yes. I can also do the same cross here. I can simply cross it like this. But that would be so confusing in the end. It's better not keep it confusing. You can simply draw a square and make it easy. And it looks good too. In your answer sheet, it would look good. Yeah. So just try to learn this. And from this, we can easily come to the conclusion that here the phenotypic ratio here is, okay, once again, oh, so here the phenotypic ratio is equal to phenotypic. What is phenotype and what is genotype? I hope you remember. So the phenotypic ratio is equal to how many phenotypes are available here? How many physical expressions? How many morphological expressions are here? Tall plants and the dwarf plants. Simple, just two, right? So the phenotypic ratio is equal to tall is to dwarf, which is equal to how many tall? 1, 2, 3. So 3 is to 1 dwarf plant, right? So that is the phenotypic expression. And whenever you are writing a monohybrid cross or a dihybrid cross, always make sure that you rewrite the sentence which was asked in the question. If the question has asked you to give the phenotypic expression, at the end of this Punnett square, after doing all the cross, just rewrite the answer saying the phenotypic ratio is equal to so and so. It's always important that you rewrite the sentence and maybe underline it or maybe keep it inside a rectangle box, whatever. Just always highlight in the final answer. Don't just leave the answer paper with all the crosses done and let the teacher take whatever mark or whatever point she wants. Okay, always highlight in the answer which was asked in the question. And when we are solving some questions, I'll tell you how to do that. Okay, and in the second case, we have to find the genotypic ratio. So how many types of genotypes are available here? How many types of genotypes are available here? See, sorry. capital T. Okay, once again. Okay. So we have capital T, which is one genotype. So capital T is to capital T, small t, which is the second phenotype, is to small t. So there are three genotypes available here. Capital T, homozygous dominant. How many? Just one. So that is equal to 1 is to capital T, small t, heterozygous dominant. How many? 1, 2. 2 is to small t, homozygous recessive. How many? 1. So in the monohybrid cross, when you are studying the inheritance of one gene, the phenotypic ratio, no matter which character you choose, no matter which organism you choose, the phenotypic ratio of the F2 generation is always going to be 3:1. And the genotypic ratio is always going to be 1:2:1. Keep these things in your mind. It is very important. Never forget the ratios. Okay. This is the most important part of your monohybrid cross. Is this much clear? Any issues? Yeah. Thank you, Gummi. Yes. Yes. Thank you so much. Any issues from this part? If not, then I'm moving on. Okay. Now let's solve a question. Okay. In a pea plant, the trait of flowers bearing purple color, capital P. So it's dominant. Is dominant over the white color, that is small p. Explain the inheritance pattern of F1 and F2 generations with the help of a cross following the rules of inheritance of traits. State the visible characteristics of F1 and F2. So that is the question. State the visible characteristics of F1 and F2 generation and explain the inheritance with the help of a cross. So two things are asked here. One is make the cross, take the F1 and F2 generations, and then state the visible characteristics. So whenever you get a genetics question, whenever you get a question from the heredity, make sure what the question is asked for. Normally what we make mistake is that where the mistake goes is that we would do all the cross, we would do all the self fertilization, cross fertilization, we would get the ratio, everything, but in the end we forget to rewrite the answer and never give any attention to the questionnaire, and whoever put that question would feel so bad because the question would be asking something else and you just have done the cross, right? So whenever you get a heredity question, make sure you are rewriting the answer. Just highlight on the final answer. So here we have to get two things. One is the cross, second is the visible character. So let's do the cross very quickly. It's very simple. It's just the same one that we have just done, right? It's the same. So here the character is just different. We have purple. The letter is already given. So it's capital P into small p. That's the parental generation. You can either write parental generation or you can write parents or you can write capital P. No problem. So capital P into small p. And then take the gametes. Here the gamete would be capital P, which is crossed against small p. And in the F1 generation, you would get capital P, small p, which are all purple flowers. That's F1 generation. Now, to make the second cross, that is, to do the second cross, you have to take the parent as F1 generation, right? That's how you take it. So take the parent as capital P, small p into capital P, small p. The gametes would be, there are two types of gametes available here. We'll have a capital P, small p from one parent and capital P, small p from the other parent. Now we have to take the F2 generation. For the F2 generation, without making any confusions, I don't want any confusion, so I'm just going to make a Punnett square. Simple like that. Okay. So I just have to make a square find the F2 generation. Yes, just like how we did the tall and dwarf, I'm doing it. I hope these things are clear. If there is any query, just ask it in between. Okay. We have the male, female, capital P, small p, capital P, small p. The babies would be capital P, which is a purple flower, capital P, small p, which is another purple flower, capital P, small p, another purple flower, then small p, which should be a white flower. So the phenotypic ratio is equal to phenotypic. Phenotypic ratio is equal to purple is to white equals, purple is to white equals 3 is to 1. The genotypic ratio equals genotypic ratio equals capital P is to capital P small p is to small p equals 1 is to 2 is to 1. Simple. Now, you can end the question there, but what was the question asked for? What was the question? Make a cross. Done. State the visible characteristics of F1 and F2 generation. Now that's the question. You have to make the visible characteristics of F1 and F2 generation. So you have to rewrite the question in the end as visible characteristics. Visible characteristics of F1 equals. So visible characteristic means a phenotype or genotype. It will be the phenotype, right? So here in F1 generation, the only visible characteristic is purple. So in the F1, the visible characteristic is purple flower. That's what you have to rewrite. This is where you can lose your score. Rewrite it. Then visible characteristic in the F2 generation. Visible characteristic in or of F2 generation is equal to. So let's see, in the F2 generation, how many characters are available? We have purple, 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 white. That is purple and white. Yes, purple and white. And if needed, you can also rewrite the ratio, no problem. It's 3 is to 1 in the brackets, no problem. Additional pool. So this is how you have to answer a question when you get something from the cross part. Okay, if they're asking to make a monohybrid cross, this is how you have to answer it. Simple like that. Clear? Clear? Now this is another kind of question which is very simple but from the same topic. From the same topic, monohybrid cross. This is another question but very simple. You can answer it just like that. Two pink colored flowers on crossing resulted in one red, two pink, one white flower progeny. The nature of cross will be, double fertilization, self-pollination, cross-fertilization, no fertilization. So what is the nature of cross? What is the nature of, what is it? It is a crossing. What kind of cross is happening here? Is there any guess? From the question, it is so simple. So simple that two pink colored flowers are crossed and we are getting 1 is to 2 is to 1. So where do you get the 1 is to 2 is to 1? Definitely not in the F1 cross, right? Definitely not in the cross fertilization. You won't get 1 is to 2 is to 1. Only in the second cross, that is self fertilization, you will get self pollination or self fertilization, you will get the 1 is to 2 is to 1. So it's nothing other than self pollination, right? It's a self pollination between two pink color flowers. Okay, that's it. That's pretty much about the monohybrid cross. I hope everything until now is crystal clear to you. Okay, now let's quickly go through the dihybrid cross. Now we have explained the monohybrid cross in such a way that I can easily go through the dihybrid cross. There won't be much issues here. It's the same thing but just with two character. So inheritance of two character is studied using a dihybrid cross. Here also there are two steps. First, cross fertilization and then self-fertilization. But this crossing and selfing would be little difficult because we are using two different characters. Okay, so here I am taking two different character. One is the seed shape and the other one is seed color. So I am choosing two character, seed shape and seed color. Okay, seed shape and seed color. So let's see how it's gonna be. Okay, you ready for the dihybrid cross? Okay, so the seed shape, as you already know, we have two types of seeds. We have the round seeds, which is a dominant trait, and wrinkle seed, right? We have round seeds and wrinkle seed, round and wrinkle, two different traits. Then we are choosing seed color. There are two different seed colors possible. One is the yellow seed and the other one is green seed. And which one is dominant? Which one is dominant? It is the yellow seed. Okay. So we have the yellow seed, which is the dominant trait. And then we have the green seed, which is the recessive trait. So we are basically taking two different alphabets for it. Round is dominant. We are taking capital R, capital R. Wrinkled would be small r, small r. Always take the letter for the dominant trait. Here, yellow is the dominant. So we are taking capital Y, capital Y. And for green, we are choosing, yeah. For green, we are choosing small y, small y. Small y, small y. Okay, this is how the letters are taken. Now let's see how the cross is done. So we'll quickly go through the crosses and let's see. Okay. Oh, sorry. Okay. So the cross is going to be like this. The parents are round yellow seeds and the parental generation is going to be round yellow seeds, into which is the recessive ones here. The recessive one is wrinkled green seeds. Okay, simple. So the parents are capital R, capital R, capital Y, capital Y. Into, against, small r, small r, small y, small y. Right? So the gametes here are going to be a little different. The gametes are going to be little different. You will get capital R, small y, one gamete. One gamete will receive both the characters allele. One for the shape, one for the color. Here, the gamete is going to be small r, small y. And in the F1 generation, the first generation, how is it going to be? The first generation is going to be like this. That is capital R, small r, capital Y, small y, which is a round yellow offspring. Round yellow offspring. So, all the plants which you get from that cross fertilization will be round colored seed and yellow, round shaped seed and yellow colored seed. Okay, so all of them will be round yellow. Now let's go through the F2. Let's do the F2. Since F2 is a bit harder one here, I have already started the cross. Like you will make the what? You will have to make the F2 generations from these gametes, right? You are going to self-fertilize the F1 generation. So I'll show how you are crossing it. Okay, I'll use another color. Okay. So the parents are going to be capital R, small r, capital Y, small y against capital R, small r, capital Y, small y, right? Now the gametes would look like both of the parents would give the same kind of gametes. So let's see which all gametes are possible here. It's not just one. You have four different alleles here. So you can get four different kinds of gametes. First would be capital R, capital Y, one possibility. Then there is another possibility of capital R, small y, second gamete. Small r, capital Y, another possibility. Small r, capital Y. Then you have small r, small y, the next possibility. Got it? Got it? So these are the 4 types of gametes which would be available from these parents. Okay. Now we are going to make a self-fertilization which should give the babies, right? So let's see how many types of babies would be there. I've already created a Punnett square here. It would make our job easier, right? Okay, so in the Punnett square, there will be one male, one female, and these are the gametes. Capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. It's the same for both, same for both. Now, when I'm making a cross for the first row, I'll simply make a cross and you'll understand. The remaining I've already done. So this would be capital R, capital R, capital Y, capital Y. See, capital R, capital R, capital Y, which will be a round yellow plant, right? Round and yellow plant, not plant, the seed. A plant with round yellow seeds. Then capital R, capital Y, small y. Capital R, capital Y, small y. That is round yellow seed. Round yellow seed. Same thing. I hope you're understanding. I hope you're following. It's the same thing. We are just repeatedly saying the same thing. Hi. One and only. Hi. One and only. Okay. Then we have capital R, small r, capital Y, capital Y. Again, round yellow. Round yellow. Then we have capital R, small r, capital Y, small y. Again, round yellow. Got it? And that's how you can finish the entire column. You can see capital R, capital Y, small y, capital R, small y, and such that in the same way you can complete the entire column, from which we can totally understand very easily which all possibilities are there in the F2 generation. So this is the F2 generation, right? And when you are making an F2 generation phenotypic ratio, the genotypic ratio is going to be very long. It's going to be very long, which is not at all needed for us. Normally, there won't be any, there haven't been any question from the genotypic part, because there would be 9 types of genotypes here. There would be 9 types of genotypes from this, which is little complicated. And you need to memorize this 9:3:3:1, which is the genotypic ratio here. So from this column, if you sit and count, you can get 4 types of phenotypes. 4 types of phenotypic expression. One is red, yellow, which are the dominant traits. Then wrinkle green, which are the recessive traits. And 2 intermediates, the round green and the wrinkled yellow. The round green and the wrinkled yellow. Yes. Hi. Okay. Hi. Since none of you are in your real names, I cannot even say hi. So hi. Yes. Trust me, one and only, all, everyone. Hi, hi. I hope you all use your own real names so that I can at least say your name. Hi. Okay. So, okay, fine. So, this is how the phenotypic ratio is taken out of the dihybrid cross, that is 9:3:3:1. If you remember, in the monohybrid cross, the phenotypic ratio was 3:1. Keep it in your mind. And genotypic ratio is 1:2:1. Simple. Here in the dihybrid cross, the phenotypic ratio will always be 9:3:3:1. Keep it in your mind. These ratios, very important. Keep it in your mind. Yes. Okay. Okay, fine. So, I think we can quickly do a question from the dihybrid cross and maybe wind up the session. I thought I'll be able to wind up the session today, but no. We have much more topics to cover up. We'll do it in the upcoming class. So, we'll wind up the class with some question from the dihybrid cross. Okay. So, let's see what the dihybrid questions are. So, first one. A Mendelian experiment consisted of breeding tall pea plants bearing violet flowers with short pea plants bearing white flowers. I hope the question is clear. So here is some sort of breeding which is between two different characters, right? We have tall pea plants which are giving violet flowers. So tall and violet is the one character. So we are crossing tall violet, tall plants which give violet flower with short pea plant bearing white flower. Short pea plant bearing white flower. So that will be short white. That's the cross. Okay. The progeny all bore violet flowers. So when this cross was done, all the progeny in the generation had violet flowers. So from there itself we have got a clue. All the progenies are violet means there is already a clue in the question. There is a hint. But almost half of them were short. Okay. Half of them were short. So all the progenies are violet, but half of them were short, which means half of them were tall. There were tall and short in equal in equal ratios. This suggests that the genetic makeup of the tall parent can be depicted as. Okay, so now we need to understand what is the genotype of the tall plant. The parent plant, which is tall, we need the genetic composition, the genotype of the tall plant. Got it? Okay. So, yeah. With this answer, we'll be winding up the session. If you already know the answer, the red ones are better. You like violet flowers? That is not our business, dear students. The flower being red and purple is not our business. Our business is the answer. We have to choose one of the options so that you might get 1 mark here. Please focus. Okay, fine. Okay, so we have the tall violet plant and the short white. We got all the progeny violet. All the progeny are violet. So I need the genetic composition. Simple, very simple question. It might look very confusing, but simple. So if the plant is tall, there are two possibilities. Capital T or capital T or capital T small t, right? Or capital T small t. That's one possibility. Then all the flowers are violet. If all the flowers are violet, then the possibilities are capital W or capital W small w. Both are possibilities. Right? Going to the short. Short, it's definitely small t, and white, it's definitely small w. That's the only possibility. It is homozygous recessive condition. But here we have to choose which genotype is the correct one. It's a very simple question. See here. They have already said we got a violet progeny and not one violet progeny. All the progenies formed here are violet. If all the progenies should be violet, then it should be a dominant trait, which means this parent should give one capital W from here, and that parent should always give one small w, right? So only if this tall violet parent is giving capital W, then we get a violet progeny in the complete generation. In that first generation, all the violet flowers should be violet. In the first generation, it all should be violet. Only if you get capital W and small w, right? If you get a small w here, if you get a small w from this parent, then it is going to create small w baby. That is a white baby, not violet. So this possibility is not at all possible. This capital W, small w is not possible, because if it is capital W, small w parent, the small w and this small w will give a white flower, which is not possible. So definitely this can be cut out. So the parent is going to be capital W, capital W. What about tall? What about tall? We have already gotten the answer that half of them are short. Half of them are short, which means the other half will be tall, right? If half of them are short, the other half should be tall. So it's definitely a heterozygous character, because already we have small t in that parent. Only if there is at least one t here, then we'll get a short and tall in equal ratio. It cannot be homozygous tall. You can do the cross and find it. Let's see. If I'm crossing capital T and small t, all the babies I'm going to get will be capital T, small t ones. I won't get the short plants. But if I'm crossing capital T, with small t, then there is a possibility that I might get, see, capital T, and small t, which is 1:1. Half of them are tall, half of them are white. Sorry, half of them are short. Half of them are tall, half of them are short. Simple like that. So it's definitely capital T, small t. Only if the parent is capital T, small t, then there is a possibility of this particular half short and tall. So the progeny, this tall violet plant should be capital T, small t, and capital W. That is option C. The question looks very simple, very small, but this is how you can come to a conclusion. I hope you understood it. And with that point, I'm winding up the session. I hope we all will meet again to conclude the chapter in the next class. Please come back for the live class. I'm hoping we all will be seeing each other again. I hope there are no doubts. Here. Yeah. Yes. Hi. Okay. Why do we all look like our parents? Why do we resemble our parents? Eva, you were not in my other class. No, you were there in my other class. I think you didn't attend my class very carefully. Yeah, she didn't attend our class very carefully. So it's simple. Why we resemble our parent is because we are getting the same genes. We are getting the same genetic constitution from our papa and our mama through the sexual reproduction. Whenever there is a sperm carrying the genetics from the father and an egg carrying the genetics from the mother, and when these two fuse together, the zygote is formed, and the zygote is us. So definitely we have all the characters of our papa and our mama. That's why we resemble our parents. Simple. Yes. Okay. Okay. Fine. So with that, I think we are good enough to conclude our session. I hope everything is fine. Yes, I would like to see each and every one of you again for the last session to wind up this heredity. And thank you so much staying back with us in the entire session. Thank you so much for all the good comments. Thank you so much for all the love and support. And we'll see each other in the next class. Take care. Goodbye. This is Alka Rajimon from GoSco. Good night. Bye.